Hello, and thank you for joining us for today's webinar, OCT Evaluation of Neovascular AMD in Clinical Trials, a Reading Center Perspective. My name is Amanda Weber, Partnerships Manager at Merit. For those of you who are attending for the first time, we're delighted that you could join us. And of course, we extend our deepest appreciation for all returning attendees as well. This presentation will provide an overview of the role a reading center plays in the use of optical coherence tomography in major studies for patients with neovascular age-related macular degeneration, reviewing the capabilities of the reading center, as well as summarizing how these studies are approached. If you are joining us live for the first time, you may also view Merit's previously hosted webinars on our website at meritcro.com. Under the Resources tab, select Webinars to replay your on-demand webinar of choice. We also host this content on our company's YouTube channel. Please note we are recording this webinar so that it can be added to both our website and YouTube channel for replay. All attendees will receive a follow-up email, and that will contain a link to this recording, which we welcome you to share with fellow colleagues and industry connections as you see fit. All participants will be muted during the presentation. However, we will be devoting time to questions and answers at the end. We invite and encourage you to submit questions at any time throughout today's presentation via the Zoom Q&A functionality. We will address as many questions live as time permits, and then all remaining questions will be answered directly via email following the webinar. Without further ado, I would like to introduce our webinar panelists and speaker. Dr. Ron Danis serves as Merit's co-founder and chief scientific officer. He brings extensive experience to the company as a practicing medical retina specialist. Dr. Danis is involved in the design and conduct of many clinical trials and serves on leadership committees as well as data safety and monitoring boards for several ongoing studies. Dr. Joe, Merit's VP of Medical and Scientific Affairs, oversees Merit's team of graders, data scientists, and analysts. She works extensively with study sponsors and independent reading centers to develop imaging protocols and grading forms to meet the specific needs of each trial, while maintaining quality initiatives to ensure effective clinical trial execution for our partners. And our speaker for today, Dr. Shawn Wilker, Merit's Medical Director of Ophthalmology. Dr. Wilker is board certified in ophthalmology and a retinal specialist with over 15 years of clinical experience. At Merit, he spearheads the development of evaluation procedures for ophthalmic imaging grading. His roles include analyzing and reading images and/or data for ophthalmic clinical trials, performing reading quality control tasks, developing analysis and reading procedures, as well as providing training for readers. Dr. Wilker, we're looking forward to the content you've prepared for us today. You now have the floor.
Thank you, Amanda. I appreciate that. And I'd like to thank you and welcome everybody this morning for joining us. So OCT is one of the most commonly used modalities when evaluating the retina. And this is regardless of whether you're talking about in the clinic or whether you're talking about in clinical trials. So today we're going to focus on the use of OCT in clinical trials specifically related to patients that have wet macular degeneration. In this presentation, I'm hoping to give a kind of a start to finish methodology of how we approach using this modality in the setting of clinical trials with wet macular degeneration. I'm going to go over some of the more commonly used metrics that we have seen from the different sponsors. I'll talk about some of the limitations of OCT as a standalone modality. And then I will touch on just a few of the more experimental or less commonly used metrics that we have seen from time to time from different sponsors. As I've already touched on, OCT is rarely used by itself. It is oftentimes paired with at least a fluorescein angiogram, at least a colored photograph, very rarely ICG, and autofluorescence is becoming more common. We're going to focus, however, only on OCT, and I'm not going to get into OCTA, which will likely be covered in a future talk. I've given some definitions here. This is really for your reference later. We will not go over them at the moment. So before the study even begins, there is a tremendous amount of work that needs to happen. There are a variety of different commercially available systems out there. Each one of these needs their own protocols, scan settings, how to acquire them. Our team has to work with the individual sites to make sure that they have a photographer that's able to acquire the images that we require, as well as doing so with good quality. And then after they are able to get those quality images, they need to be able to upload them to our systems. Over here, I've given some examples such as Heidelberg Spectralis OCT, as well as Zeiss Cirrus and Topcon. These are some of the systems that we have used in the past, but this is by no means an extensive list. And as new systems come online, we're usually able to incorporate them into our system. So here is where I would like to introduce you to our platform, which is called Excelsior. Excelsior is one validated secured system. It contains all study data, images, tasks, queries, site materials, analysis, and the data that is read. It is basically a one-stop shop. It's very user-friendly, and we usually get high accolades from the sites when using this system. It's cloud-based, so we don't have to worry if an individual site is having some sort of an IT problem. It allows for us to compare these different commercially available OCTs, and we do that through the use of the DICOM standard. It is also a 510(k) cleared medical device for ophthalmic and radiological indications. It's HIPAA compliant, and it meets all of the criteria that are needed for electronic records and signatures. So here I'm going to start giving you some examples of what we see when we look at these OCTs while we're working with Excelsior. So here what we have is one of the B scans that I'm currently reviewing, but up in this corner, I'm able to scroll through all of the B scans. In this particular scan, they've acquired 97. I can scroll through and see how one B scan relates to another. Beyond that, I can start doing split screens. So I can take a look and see, how does this compare to a previous scan? So over on this side of the image, this was visit 1, and now I can look over here at visit 6 and see what type of changes have occurred and make comparisons on our grading forms. Also, if I want to pull in one of those other modalities, such as here, where we've got a color photograph of somebody with a disc-formed scar and some RPE changes, I can now correlate that to what it looks like on today's OCT. The next thing that we oftentimes do is what's called segmenting. So the retina is made up of a multitude of different layers, and we can individually outline each one of those, and that gives us our different segmentation. And then through any sort of combination of those, we can come up with different measurements. And here we've given an example of this. In this example, we've got the ILMs marked up here, as well as the RPE. And here we're able to come up with different types of measurements. We'll talk about that much more in the presentation. Beyond that, we're able to answer morphology questions. How reliable is the scan? Can I actually read the scan? What type of things am I seeing? Am I seeing things like subretinal fluid? Am I seeing pigment epithelial detachments? And then one nice feature is we have this ability, once we have segmented this into different types of layers, I can then use it, I can turn it into a map, and I can give this kind of en face image that I can use to give a topographic kind of an image to tell what the macula looks like. So in this example, again, where we have the RPE segmented, you have these elevation of the drusen here on the RPE. And when I look at it and en face, I can see where the drusen lie throughout the macula. Another nice feature about that is once I have segmented and I have my en face image, I can actually make measurements. So here we have an area indicated by the blue arrow of Cirrhoa. And when I look at the en face image, I've got my area now of geographic atrophy, and I can make a number of different measurements with this. So at the beginning of the study, we are usually tasked with the eligibility, which includes who's going to be allowed in or who's not going to be allowed into the study. So what I've done here is I've given on this side of the page just kind of an example of what we might get from the sponsor. They have typically come to us, they already have a protocol. Within their protocol, there are usually different types of imaging modality criteria, and we take that and we turn it into what we call our reading center charter. And this is basically our roadmap of what we are going to grade and what we're going to be looking for when we look at these different patients and subjects. So as you can imagine in eligibility, if you're talking about a wet macular degeneration study, the first question always is there actually wet macular degeneration? So as I've already touched on, multiple modalities are typically used. Over here, I've got a fluorescein angiogram, but over here, I'm going to touch specifically on the features that we're looking for in the OCT. So the first is supporting features that show that this person has macular degeneration in the first point. So these yellow arrows are currently pointing to these small elevations of RPE, which correlate to drusen. Other things we're looking for, we're talking about wet macular degeneration, which is an exudative process. So we're looking for signs of exudation, such as subretinal fluid here indicated by this hyporeflective wedge, but we'll also be looking for other signs of exudation, such as CME. Pigment epithelial detachments, which are larger elevation of the RPE, are also things that will help to support the fact that this person has macular degeneration in the first place. And then another feature, which we will touch on a little bit more later, is something called shroom. Shroom is subretinal hyperreflective material, and that is a bright area or a bright hyperreflective area that goes between the RPE, which I've got outlined here in the blue, and the retina up here. So inside that subretinal space, there's a hyperreflective material that can mean a few different things that we'll talk about later. Morphological things that we are usually asked to look for when we're talking about eligibility, one of the first things is macular atrophy. And here I have given the CAM definitions. CAM stands for the Classification of Atrophy Meetings. And these are the 4 standard definitions that are used by the CAM to identify macular atrophy. One of the most common things that we are asked in a wet macular degeneration study is, does that person have geographic atrophy or macular atrophy that involves the foveal center? And this makes sense for why this happens. If you have a medication or a treatment that you are hoping is going to actually improve a person's vision, then the last thing you want to do is enroll a subject that has another confounding issue that's going to limit how good that vision can become. While these are the standard definitions, we are occasionally asked by different sponsors to veer from these definitions. The most common one of the definitions that we veer from is sometimes the sponsor wants us to use a lower number than the standard 250 micrometers. The next thing, which kind of goes along the same vein, is the last thing the sponsor wants is subretinal fibrosis in the center. So here you have a hyper-reflective area, this kind of a dense, thick scar that has occurred. And again, you're in the same kind of a situation where if you enroll a subject in a study like this, and they already have something like subretinal fibrosis in the foveal center, then even if the treatment works, that subject may not be able to have an improvement in their vision, and then that affects the end results. The other morphology question that we are very frequently asked about when we're talking about eligibility is an RPE tear or rip. Here I've given an example of an old tear where you can see this hypertrophic band here and this kind of hypopigmented area over here. What has happened is the RPE has actually separated and ripped, and it's scrolled up here. In the early setting when this happens, this is very hypopigmented, but over time it can get a little bit more pigmentation because of its scarring. But this is what it looks like on an OCT. This yellow arrow indicates the scrolled edge. So this used to be over here and now has torn and is bunched up over here. And then you can see these undulations here of the RPE almost looking kind of like an accordion. And the thing very similar to a subretinal fibrosis or macular atrophy, patients that have these can limit the ultimate visual potential, and sometimes they can even limit how good the treatment is itself. And so we are often asked to use this as an exclusion criteria. Beyond the morphology things that I have talked about, we are occasionally asked, even in the eligibility portion, about measurements. This will become more common when we get into the grading, but there are some sponsors that want us to start off with the measurements depending on what it is that they are looking for. The most common by far is the central subfield thickness. Again, I've got an individual that has been segmented for the ILM and the RPE, and we're looking for the thickness in between these two measurements, but we're trying to do it in the central subfield. That by far is the most common measurement that we're asked about. The measurement that goes kind of hand in hand with this is called the center point thickness, which is really just that one single point in the dead center. Beyond that, less commonly, we are asked about volume, but it is usually put in there as kind of an extra measurement. And then the other thing we are sometimes asked about in measurement is more, again, along the exclusion criteria. Pigment epithelial detachment height is sometimes put in there because sponsors don't want a subject that has a very large pigment epithelial detachment. The fear there is that pigment epithelial detachments have a bad habit of collapsing. And when they collapse, they can turn into atrophy. And again, you get into that same kind of a situation where you have a limitation of vision or even a worsening of vision during the treatment with the medicine that or treatment that they are studying. So now that we've decided who is going to be eligible for the study, then we get into the heart of the study, which is the grading. We start off in a very similar fashion. When the sponsor comes to us, they oftentimes have already got a protocol that has identified their primary and secondary endpoints. And whatever it is that is what they are looking for is how we tailor our charters. So here I've given just some examples. We'll go through a lot of this in more detail, but this again is our roadmap for how we're actually now going to grade these individuals or subjects that are in the study. These are the common questions, and we're going to go over this also in more detail. So almost always the very first question is this. We're almost always asked about center subfield thickness and center point thickness. So as I've already touched on, we segment into the ILM to the RPE, as you can see here by these green lines. And the ETDRS grid is centered here on the foveal center, as you can see here. And then the center subfield thickness is the average measurement of the thickness of the circle with this 1 millimeter diameter centered on the fovea. And as I touched on earlier, the center point thickness is that single point measurement right at the center. Beyond the measurements, we start getting into different morphology questions. So it is very common for us to be asked, is there subretinal fluid, as identified here by this hyporeflective wedge underneath the yellow arrow? And we're oftentimes asked, if there is fluid, where is it? The most common of these questions is it in the center point? So here you can see in our ETDRS grid, there is the crosshair here, which is in the center of the fovea. This correlates to this line here and shows us that, yes, there is in fact subretinal fluid in the center point. Beyond that, we are oftentimes asked to measure this, and this is something that they will follow over the course of the study. Sometimes the sponsor doesn't care as much as if it is in the center point, but where is the largest amount of subretinal fluid? So in this example, the crosshair is now in the inferior outer subfield. And you can see here we've made a measurement of the maximum height of subretinal fluid that this particular subject has. A less common measurement, but one that we do see occasionally is us being asked, rather than measuring the height, to actually measure the horizontal width. This is kind of a corner to corner measurement. And we've seen this in a couple studies, but I would not call it that common. Next would be intraretinal fluid. So intraretinal fluid is oftentimes seen by cyst in the retina. Again, the same types of questions occur. Where is that intraretinal fluid? Is it outside of the center subfield, as in this example? Or is it in the center subfield, as seen in this example? We sometimes, but not often, are asked to identify more characteristics about the edema. So in this example, in which you see a lot of these dark circles, this would be called cystoid macular edema. But this is compared to this patient in which they have this hyper-reflective area here, which indicates a scar. But over here, you have the retina, which is clearly thickened, but it has not devolved into these cysts the way that this example. So we are sometimes asked to identify whether it's cystoid versus non-cystoid. Moving on to morphology for pigment epithelial detachment presence. Again, the same type of questions are asked. Is it outside the center subfield or is it in the center subfield? We a lot of times are asked about different types of morphologies. There are the three common ones: serous, fibrovascular, and the less common one, hemorrhagic. A serous pigment epithelial detachment is this elevation of the RPE above Brooks membrane, and it has a very uniformly hyporeflective or dark center. It also tends to be evenly shaped. It has this nice kind of a round architecture here. And this is in comparison to the fibrovascular pigment epithelial detachment, which also has that RPE elevation above Brooks membrane, but there's all kinds of differences in the hyperreflectivities within that area. And the more hyperreflective bands that you have, more likely to have more fibrosis inside of that pigment epithelial detachment. Unlike the serous pigment epithelial detachments, these are not round and they are not evenly shaped. They tend to have a lot of peaks and plateaus. And then the least common one that we are asked about is a hemorrhagic pigment epithelial detachment. And this is where you've actually had a hemorrhage underneath the RPE. And depending on how bad the hemorrhage was, you can get a variety of different types of hyperreflectivities or hyporeflectivities underneath the dome. So here, these arrows are pointing to subretinal fluid, and this dome is outlining the RPE over this area of hemorrhage. Hemorrhage is very reflective on an OCT, and what happens is as the light source comes through, it's very bright on the surface as it's reflecting from the blood, but the signal cannot penetrate deeply, and so you get this hyporeflective area. This is not because there is fluid like serous fluid underneath here, but rather it is an artifact of the fact that the signal cannot penetrate deeply enough into this pigment epithelial detachment. And then beyond that, all kinds of things can change as the pigment epithelial detachment resolves. Going on to measurements for pigment epithelial detachments, probably the one that we are oftentimes asked about, again, is thickness at the center point, but we can have similar measurements to some of the other morphology features, such as the maximum thickness. And very unusually, we are asked about this maximal horizontal distance of a pigment epithelial detachment. And then moving on to morphology about the neovascular lesion complex, we get into some of the same kinds of questions. Where is it located? Is it in the center subfield? Is it outside the center subfield? And then we are asked about the type of lesion that we're dealing with. So there are three different types. And while they were born out of the fluorescein angiogram era, there are now ways to define these different types of lesion using the OCT. So the first of these is type 1, which is also known as an occult choroidal neovascular membrane. This is one that arises out of the choriocapillaris. It penetrates through Brooks membrane and resides in between the RPE and the Brooks membrane. So this is what is known in the literature as the double layer sign. So the double layer sign is this. The red arrow here is pointing to the hyperreflective band here, which is the RPE, and the green arrow here is pointing to the hyperreflective area, which is the Brooks membrane. In between this, there is still some hyperreflectivity, but it is less hyperreflective than the red or the green arrows. This is where the choroidal neovascular membrane is thought to be residing. The second type is type 2, and this is associated with a classic choroidal neovascular membrane, which evolves out of the choroidal vasculature. It transverses the RPE Brooks membrane and ultimately ends up in that subretinal space. So this is the example I used earlier, where the green line here is Brooks membrane. This blue line is the RPE, and you can see this is a pigment epithelial detachment. These hyporeflective wedges here are subretinal fluid. And now in this green area, you have a hyperreflective area right underneath the fovea. This is what is called shroom. Shroom can mean a choroidal neovascular membrane. However, there are times that shroom actually indicates other type of hyperreflective materials such as scar tissue or blood. And then the last type of choroidal neovascular membrane is type 3, and this is the least common of the different types of choroidal neovascular membranes. This is oftentimes spoken about as a wrap lesion. This is one that the idea is, rather than starting down here in the choroid, the idea is that it actually develops first in the retina itself and works its way down. On an OCT, you can see some cystoid macular edema. As this thing progresses, it will eventually break through this complex. And so in that point, you can start to get different types of either serous or vascularized pigment epithelial detachments. I find these difficult to determine on OCT by themselves. So now we've finished with our grading. Then we get to the post-grading, and there are some final steps that we do before we ultimately give this data to the sponsor. One thing that I have not touched on much up until this point, but I will touch on a little bit now, is something that we call workflow. Workflow is, how is this individual subject going to move through our system? There are a variety of different ways that we do this, and it really is tailored to what it is that the sponsor needs. So an example of this would be, how many graders are going to look at this before we call this a complete reading? You might have some sponsors that only need one person to grade it, and that's it. Then there's others that want 2 or 3, and they want a variety of different ways for this to be handled. They might want complete agreement between the different subjects, or they might want a tiebreaker. So there's a variety of different ways that we can handle this based on what it is that the sponsor needs. We also do a lot of different quality control steps afterwards, in which we run the data. We make sure that human error is brought to a minimum. We look for signs of things like misclicks or measurements that don't make sense, and we go back and we review that to make sure that we are giving quality results to the sponsor. And then finally, we actually do the data transfer and give the information over to the sponsor and allow them to do their own analysis and data mining there. Once that is finished, oftentimes there are still questions that need to be answered by the sponsor, and we're available to help them interpret the data that we've given. So what we've done here today, we have reviewed the Reading Center's perspective on regards to the use of OCT and the role of wet macular degeneration studies. And while I have specifically talked about OCT and I've talked about wet macular degeneration, the methodology and the backbone that I've given here really can be extrapolated to other modalities and to other types of diseases. So I've given kind of an idea of just how it is that we function as a reading center. But within this, I have gone over the overall process involved in eligibility and grading of wet macular degeneration with use of OCT. I've highlighted some of the capabilities that we have using Excelsior and bringing multiple commercially available OCTs from different manufacturers. And I've talked about some of the common measurements and questions, endpoints, and morphological features used by the different sponsors in this trials. So at this point, I am actually going to hand this back over to Amanda. I'd like to thank you all for your attention, and I believe that we are going to move into our question portion.
Yes. Thank you, Dr. Wilker, for that very informative presentation. We have reached the Q&A portion of today's discussion. If you do have any questions, please submit them now via the Q&A functionality. Prior to addressing your questions, I would like to share just a brief company overview. So Merit is an innovative global clinical trial endpoint services provider focused on a variety of therapeutic areas while specializing in ophthalmology, respiratory, and oncology. And we've served biotech and pharmaceutical companies in their clinical endpoint analysis for over 10 years with a team of medical professionals as well as seasoned operational support staff. Positioned as your clinical endpoint expert, Merit provides very comprehensive endpoint assessments powered by our proprietary single system software platform that was designed to manage the entirety of the workflow process, from acquisition directly from the sites, storage, quality reviews, and expert analysis of that data to be then exported and returned to the sponsor, all within a very collaborative, partnership-driven environment with customized solutions and personalized attention. We will now open the discussion for any questions that have been submitted. There's a question asking if we will be able to provide the slides. They were very informative, and absolutely. We are recording this webinar, so all attendees will receive that email with the recording link so that you can re-review the slides and the content. And I'm sure we'd be able to reach out directly via email as well, perhaps with a PDF version of the slides. All right. Looks like, Dr. Wilker, we have another question. What are your thoughts on the use of AI for the interpretation of OCTs?
So AI is clearly on the forefront of a lot of people's minds. It's very common not only in medicine but outside of medicine. In the world of ophthalmology, there is a lot of research going on right now with AI, specifically with diabetic retinopathy, systems that are able to count microaneurysms or give kind of a diabetic retinopathy a severity score. In the macular degeneration world, the things that are looking for subretinal fluid or some sort of like home test so that you can identify patients that are having a change in the status of their macular degeneration. So there are definitely some good systems out there, and we've seen some good results out there. AI currently is not FDA approved to be the end-all be-all for any particular study. So that's the first point. But beyond that, the AI, while it is definitely improving, and I think that it will continue to improve, we have not yet gotten to that point that it is flawless. So you still need a human's eyes to look at something to make sure that that AI has called something the correct thing. AI can be helpful kind of as a supplement, but right now it does not supplant a human's need for being there and interpreting the data.
Absolutely. Thank you for that. And I do want to allow any speaking time or additional comments complimentary to Dr. Wilkers from our panelists as well. Any other comments on the AI from Dr. Joe or Dr. Danis?
No, I think we're all interested in learning how AI can make us more efficient at the reading center. And I have no doubt that as technology advances, we will be using AI to supplement our human graders. As Shawn has mentioned, I do think that it's a ways off before we have AI reading centers that are going to be doing clinical trial work.
Yeah, I would agree with that as well. I think that the AI definitely has a role, especially when you come to some quantitative evaluation, or that AI algorithms already incorporated into reading center activities at the different levels. But the readers still are the final kind of a gatekeeper for whether that result is ready to go or not. So I think it's going to be eventually also needed. They will be complementing each other, I think, different strengths.
Thank you, Dr. Joe. Dr. Danis. Okay, we have another question. Are there specific standards that OCT images must meet regarding quality? And how can quality control measures be effectively implemented for OCT imaging?
I actually think Dr. Joe might be the best person to answer this question. This is definitely along her line of expertise.
I think first of all, at the Merit, we have imaging procedure documents. First of all, specify the specific type of OCT images must be provided for each device, because devices are not completely uniform out there. So we're finding the type of scans and the quality requirements that's tailored to each device, but yet it's still relatively uniform across these different device provided data. So the data, the final analysis coming out of Excelsior can be pulled, the analysis can be performed in a uniform fashion. That's that. And then we providing the device and technician certification service. So we make sure that people under the instruments are meeting the requirements. And then for each submission, with each of the OCT submission, we have our imaging quality control specialist to review each submission to make sure it's meeting our requirements, first of all. And if they do have an issue, we send a query, the sites or the technician to have a chance to either collect the data or make sure in the future, similar issues do not occur. So these are the things we put in place. And then when we come to the reading side, we do evaluate because they are subjects and all that, the image just cannot be better quality, just that's the best quality it can provide. So we still based on, the evaluation needed, the grading questions and all that, gauge if the data giving us can provide, is it sufficient or not for performing the grading. So we gave a confidence score for each of the images submitted. So that's the part I think of based on the OCT quality. Ron and Shawn, maybe you guys have like to add.
I would like to add one thing. So during the presentation, I touched on the fact that in Excelsior, we typically use the DICOM standard, and that is by far going to be able to give us the best ability to make measurements and to interpret the different modalities, whatever they are, regardless of whether you're talking about OCT or something else. The nice thing about Excelsior, though, is that it is very flexible, and there are occasionally circumstances in which the sponsor is not able to provide us with DICOM images. We still can manage those. We may not be able to give every single measurement that we can do on the DICOM standard, but the nice thing about Excelsior is that, number one, it's constantly evolving, it's constantly changing. We're able to integrate other systems into our platform. And even when we are given something that is not DICOM standard compatible, we are still able to provide data analysis for those subjects.
Yeah, I might add another interesting element of this. Shawn has mentioned the DICOM standard. Why do we need to massage these various images into a standard? And famously, the manufacturers of the OCT machines have developed different algorithms for assessing measurements. And so the same eye will sometimes have quite different measurements of different features, depending on the machine on which the eye has been imaged. And so we're able to put all of these images into this DICOM standard and apply the same segmentation algorithms so that the eye will have the same measurements regardless of the machine of origin. That's a difficult technological challenge that we've been able to accomplish quite easily.
So actually, I would like to add on to that a little bit, maybe ask a question, because we know there are devices out there, the commercial instrument, they measure retinal thickness from inner limiting membrane to RPE. That's the more common type. But there are also other commercial devices providing retinal thickness measurement from IOM to Bruch's membrane. The difference in the neovascular AMD is whether PED is included in the thickness measurement or not. So we commonly see more in our studies that retinal thickness measurement is from IOM to RPE, but there are some studies from IOM to Bruch's membrane. What are the clinical considerations behind these? Why choose one over the other? What's the potential impact? Can you guys maybe elaborate a little bit?
Well, sure. These lesions are complicated, and as Shawn pointed out, there are components that are both above and below the RPE. So if we're using the RPE as our reference, we're including some elements of the lesion, but excluding others, those that are underneath the RPE. If we choose Bruch's membrane as our reference, then of course we're including stuff that is both above and below the RPE. So again, we have quite a difference in how some lesions will measure. I actually think that perhaps the best approach is to measure multiple layers. And if we're looking at the data telling us the story of a particular eye and how it's being treated, we sometimes want to look at the different compartments of the retina because they may respond differently to treatment. You know, there's some elements such as subretinal fluid respond very quickly to treatment, but other lesions such as RPE detachments will respond slowly or sometimes not at all. And the interplay of these different components of the lesion are certainly of interest. So there isn't you know, one size fits all, and it depends a little bit on the needs of the study, the patients that are being included in the study, and how much detail the sponsor would like to get into with respect to analyzing the lesion components.
Yeah, I guess with the all images in Excelsior in DICOM format, basically we could perform the whatever's required segmentation regardless of which device that OCT is coming from, and provide one or the other measurements or provide both measurements. That's all. Thank you.
Yeah, thank you for that question. I think we have time for one more question to address from the panelists and Dr. Wilker. So in addition to OCT, what other tests can be uploaded and graded within Excelsior?
Yeah, so I have mentioned a little bit even in the presentation that there's a variety of different things that we can use. OCT, obviously, but beyond that, color photographs, fluorescein angiograms, ICG, autofluorescence is becoming very, very popular right now, especially where there's so much interest in geographic atrophy at the moment. But even some of the less common modalities such as VEP, or perhaps you're looking for different types of full stimulus fields and things of that nature, we can look at visual fields and we can look at ERGs. And then the other nice thing is, as I mentioned, as new things come on board, we can incorporate them into our system as well. I've also kind of only touched on the retina stuff, but we even have you know, other studies that are more of the anterior segment in which we're doing cell counts for endothelial cell counts for different studies, and looking at the cornea and doing kind of external photographs. So we actually have a very wide variety of things that we can incorporate into our system and give accurate measurements for.
Great. All right.
May I add one thing to what Shawn has mentioned? I think the one nice thing about the Excelsior can support all those different modalities and all that, with all the data centralized, which really offer opportunity for further down the line in the data management step, because we can then transfer the data to the sponsor with consistent uniform format across these various modalities, make it much simpler from, you know, the next subsequent data analysis from subject ID and eye and all those various information. Everything is going to be in a standardized format. So that is
yes.
one.
I was actually recently on a sponsor call, and they used the word harmonization is what Excelsior brings to the table. So I really like that descriptive term.
Yeah.
All right. Any other closing comments?
I appreciate everybody coming today, and thank you for your attendance.
Yeah. Yeah, this will conclude our live Q&A session, and you can stay up to date on additional primary endpoint related content by following our company page on LinkedIn. And please stay tuned for more information regarding Merit's upcoming webinars. Thank you once again for your attendance, and have a great day.
Thank you.
Thank you very much.